Okay then, let's start the class. In the last class what we were discussing was, we were talking about the dark reaction, and as we have already discussed, what we know is dark reaction in case of different plants, the pathway is different, right. Under the heading of dark reaction we have to talk about 3 cycles, that is C3, C4 and CAM cycle. We have already discussed C3 cycle, which was also called as Kelvin cycle, and in the last class we have talked about the CAM cycle. One of the basic difference between C3 and C4, if I ask you, kuntu point koba? One major difference between C3 cycle and C4 cycle. Similarity to ami janu, both of them are nothing but dark reactions, and so the food would get synthesized. Kintu jedi moi eta sharp difference between C3 and C4 buli ko, which difference would you speak about? We generally talk about photorespiration, right. I think we have not done C2 cycle, we will be doing it today, but in spite of that I think we have discussed that topic a little bit. Photorespiration is absent in case of photorespiration is present, but komke kar hoy. What is photorespiration? It is nothing but the C2 cycle. One basic difference between C3 and C4, C3 plants bilakot ki dekha pao ami photorespiration dekha pao, and in case of C4 plants the photorespiration is totally absent, right. Another difference you can say is something about the Cran's anatomy, which we have already discussed in the last class. Cran's anatomy ki monta se, it's a special type of anatomy where we see 2 rings of cells that surrounds the vascular bundle, and this type of anatomy could be seen in the leaves of the C4 plants, right. As far as C4 plants are concerned, we have also used a word called as dimorphism or dimorphic chloroplast, right. And chloroplast boli koalo ke liye, one type of a chloroplast present in the mesophyll cell and the other type that is present in case of bundle sheath cell. 2 cells are involved in the process of photosynthesis in C4 plants. We have marked the differences also between the chloroplast of mesophyll and the chloroplast of bundle sheath. Already I can discuss korisu, right. We have to remember the word called as R granule. If you remember, one basic difference between the 2 chloroplasts that is found in the C4, ki asile R granule is a word that is important. And finally we have talked about the PS1 and PS2 code ki present hoye ase. Today we will be talking about the next cycle of dark reaction or another pathway, that is what is called as a CAM cycle. The full form of CAM is crassulacean acid metabolism. Crassulacean, it's why is it crassulacean? Because the name of the family is called as Crassulaceae. So this is a type of pathway that is found in one specific family of angiosperms. And which family are we talking about? We are talking about Crassulaceae, right. And so the pathway is called as crassulacean acid metabolism. Some of the basic points that you have to remember about this topic, no doubt we will be writing the notes, but before writing the note, the few points to be remembered. First, if you are talking about sorry, CAM cycle, first let us start with the same statement that we always say, no, je GA cycle nohok C3 cycle to hoboi lagibo. Right? Even if we are talking about C4 cycle, we have seen that C3 reactions will have to occur. Similarly, the first statement, no doubt we are talking about, will be talking about CAM cycle. In CAM cycle also, C3 cycle has to happen. Out tat ki hobo, glucose formation hobo. Second, we have to talk about, since it's a dark reaction, so we have to talk about the use of the light reaction products. Talking about the light reaction products or the assimilatory powers, no? Ki asile ATP and NADPH2, we have to talk about the requirements. It is exactly the same. Remember, we have given a chart comparing C3, C4 and CAM. Tat likhi disilu, no? Tate ki asile light reaction or kima, we are talking about ATP. What is the requirement? Obviously, ki asile exactly the same as C4. C4 or ATP kiman uses 30. So in case of CAM also, ATP kiman use about 30. NADPH2 kiman uses 12. 12 in all C3, C4, CAM. Requirement of NADPH2 is always 12, right. Next, the third point is, last class again we have talked about something called as a scotoactive stomata. There are few stomata that opens during the night time. Stomata boli kole generally ketia open koribo during the day. When as soon as the light falls, the stomata opens, right? So, kintu kisuman stomata enukwa ase jot niki rati he open koribo, tak ami ki boli kom scotoactive. And whenever the stomata opens, at that time only carbon dioxide is fixed in the plant. Right. So, atmosphere project carbon dioxide low utilize the process to arambo hobo. This is nothing but taking in of gas and releasing some gas. So, we can use the word called as exchange of gas. Exchange of gas ko through hoy, stomata through hoy. So, what we understand, stomata jetia khula thakibo tetia exchange of gas hobo. In normal cases, stomata ketia khula thake, it's always during the daytime, and so fixation of carbon dioxide always takes during the daytime. But in a specific group of plants which has a specific type of stomata that is called as photoactive stomata that opens during the night time, atmosphere or carbon dioxide to tahate ketia lobo. Jehetu stomata to rati khule, so obviously carbon dioxide ketia lobo, rati lobo. So initial fixation of carbon dioxide occurs during the night time in CAM plants. So duta point kolu, initial fixation ketia hoy, rati hoy. Next tahator eta special stomata ase, ki stomata scotoactive. And the next point is some of the examples of this family, right? Alloy, agave, bryophyllum. I think you have heard the word called as succulent, right? So CAM cycle hunile, the first word that comes to our mind is kunbilak plants hot ami dekha pao. It is always in the succulents that we see, right? C3 examples poriso, C4 examples poriso, CAM or examples porim. Out of these 3 plants, so C3 examples or iman question aha nai. The question has been asked about CAM examples and C4 examples. C4 examples, I think you remember. Give me an example of a C4 plant. Sugarcane and maize, the most commonly spoken about, right? So, hedu type is important. I think CAM plants or example will kole, respective examples that you monoton athake, succulent is a word that you have to remember. And talking about succulent, the most commonly spoken about is always bryophyllum. Bryophyllum hawk, alloy hawk, agave hawk. I think you know the meaning of succulent now. Succulencies are thake, take ami ki boli ko, succulent boli ko, right? Since we have already discussed succulency in a morphology class, I think you all understand that term, right? So today we'll be starting with the CAM. CAM to, I mean notes are arambho korim. Okay, again, what we are discussing is dark reaction. Where does dark reaction takes place? If the question is placed like that, answer ki koba? Dark reaction kot hoy? Stromat hoy or inside the chloroplast, right? So in case of C3 plants, since we are talking about dark reaction is a part of photosynthesis. Photosynthesis kot hoy? The site of photosynthesis is called as chloroplast, right? Our chloroplast kunbilak cells or bitor thake? Which cells are we concerned with? We are talking about the mesophyll cells. So what we have seen, C3 cycle, C4 cycle, or ATM, CAM, if we compare, the one thing that we say is, in case of C3, photosynthesis occur in 1 cell, 2 cells, ne? Kiman number of cells? It's only 1 cell that is involved. And which cell we are talking about is the mesophyll. So only mesophyll cell is involved in C3 for the process of photosynthesis. In case of C4, তাত আমি কি দেখিছো, 2 cells are involved, eta whole mesophyll cell or eta whole bundle sheath cell. And what about CAM? In CAM, again we'll be talking about that statement, and the statement goes up, in these types of plants, photosynthesis is confined to only 1 cell, that is mesophyll cell, right? Okol C4 plants othe duta cell involved hoy. Baki plants bilakot ki hoy, hoday photosynthesis mesophyll cell ote hoy, right? Okay, that likhasun first. Fourth, K-L-E-I-N-I-A. K-L-E-I-N-I-A. Klenia. Okay, from these points that we have written, succulent xerophyte, word to monot rakhibo laibo, right? Discovered in generally man question aha nai, but discovered by tenike aha nai. Examples of succulent xerophytes hudibo laibo, sorry, poribo laibo. Succulent xerophytes or generally, if you talk about succulent, there are certain common examples that we always remember. For example, Bryophyllum, aloe, maybe we are talking about Euphorbia also. Hey karne, especially Bryophyllum, aloe, ebilakor upor ba Opuntia, ebilakor upor questions to aha nai. Because this is something that's commonly known, right? Because since we know that CAM cycle of first word to be remembered is succulent, and succulent are most common examples is Bryophyllum, aloe, Opuntia, etc. So, ekhini it's well known. Kintu question ki hor upor is again if you talk about this, sedum or upor iman question aha nai because it's discovered in sedum only. Right, so this is supposed to be also a very common point. So, question has been asked about Euphorbia ahise, pineapple ahise, aru sometimes Welwitschia also. If you remember, sorry, plant kingdoms we have talked about gymnosperms, and what we say gymnosperms group 2 key, the smallest in the plant kingdom, right? Why is it smallest? Because maximum of them have become extinct, right? One example of a fossil gymnosperm is Welwitschia, right? So examples kini mon rakhiba. Next, continue kora in CAM cycle. Primary carbon dioxide acceptor বা primary acceptor কোন আছিলে chart খনত already দিছিলো CAM plants or carbon dioxide acceptor PEP very good Okay, the next question from the chart. First product of carboxylation, ki asile? Primary acceptor, PEP. PEP e carbon dioxide log lagi pelai ki form kore? Oxaloacetic acid, yeah. Since we are talking about what? Xerophytes, no? I think we understand what a xerophyte is. Since we are talking about xerophyte, so we have used this one. One characteristic of a xerophyte, they have a thick cuticle. What is cuticle? The upper layer of cells above the epidermis, that is what is called as cuticle. Cuticle is present in, it's a characteristic that is present in case of stems and leaves but absent in case of roots, right? Depending on which plant we are talking about, the thickness of the cuticle varies. Mesophytes bilakor thin cuticle thakibo, jar ase. As if you are talking about xerophytes, they will have a thick cuticle. What is the function of a cuticle? It checks transpiration. Transpiration, we understand the loss of water in the form of water vapor, that is called as key transpiration. So xerophytes hoy, so since they are xerophytes, they already grow in waterless condition, and so if they absorb the water and the entire amount of water goes out, তেতিয়াহ'লে তাহাঁতৰ কি হ'ব, বেছিকৈ absorb কৰিব লাগিব, পানী এনেই নাই জেগাত. So they have some measures to check the rate of transpiration. One of the measure is the presence of a thick cuticle. So question to যদি আহে, thick cuticle is a characteristic of dash plants. It's a characteristic of xerophytes, right? Okay. Next is, ইয়াতে এটা point থাকিলে. As photoactive stomata, we understood these 2 points. I think it's clear. Coming to this one, no. Since the stomata opens at night, so initial fixation of carbon dioxide occurs during the night. This is also understood. Coming to this point, we have another statement called as final carbon dioxide fixation. What we understand by the word final carbon dioxide fixation? And why are we using this word? Final carbon dioxide fixation, what actually this statement could be used in case of C4 cycle also. Final carbon dioxide fixation mane ki likhi thoi dia. C3 cycle, right? So I think you all remember we have already said, no, if it's C4, in C4 plants also C3 cycle will operate, and this cycle where the food is synthesized would be called as the final carbon dioxide fixation. If the question is asked, where does final carbon dioxide fixation occurs in C4 plants, answer ki hobo? Now, where does, C3 cycle is final carbon dioxide fixation is also called as C3 cycle. Question 2 ahise, where does where does final carbon dioxide fixation occurs in C4 plants? Bundle sheet cell. You have to name the cell, right? Kuntu cellot actually C3 cycle to hoi ase. Reaction ko pra arambho korisile mesophyll cellor pra, right? Carboxylation reaction to kot arambho korisile mesophyll cell, kintu C3 cycle kot hoisile bundle sheet cellor. So final carbon dioxide fixation kot hoisile in case of C4 plants, it occurred in the bundle sheet cell. But since we know we are talking about a CM plant photosynthesis kot hoy, it's only one cell that is involved, so everything occurs in the mesophyll itself, right? Kintu ki hobo, din ba rati, right? That is the only difference. So yate ki hobo, initial ketia hobo rati, kintu final ba food ba glucose synthesize ketia hobo daytime. Normal plants are glucose synthesize ketia hoy, generally rati. Okay. So Cran's anatomy absent, we understand this point, right? This is also something that we have already written. Photorespiration is less. We have been talking about photorespiration up till now. What we have said, photorespiration present in C3 plants, absent in C4 plants, and very less in case of CAM plants, right? Next, since we are talking about photorespiration mane ki the plants will take in oxygen, and if the oxygen amount inside the cell increases, then obviously the photosynthesis process will get inhibited. কারণ ফটোসিনথেসিস হ'ব লাগিলে সেলটোৱে কি ল'ব লাগিব, কাৰ্বন ডাইঅক্সাইড ল'ব লাগিব, কিন্তু কাৰ্বন ডাইঅক্সাইডৰ সলনি যদি তাহাঁতে অক্সিজেন লৈ লৈছে তেতিয়াহ'লে প্ৰচেছটো শ্লো ডাউন কৰিব নকৰে, ৰাইট? চ' দিছ ইজ দ্য মিনিং অফ দিছ ষ্টেটমেণ্ট। এণ্ড আগেইন চিন্স উই আৰ টকিং এবাউট জেৰ'ফাইটছ, ট্ৰান্সপিৰেশ্যন কি হ'ব, লেছ হ'ব, ৰাইট? নাও উইল ড্ৰ দ্য চাইকেল। What is Cran's anatomy? Last class में present कि absent? C4 cycle? Last class क्या था? सच्ची का क्या था? नहीं, photosynthesis का last class क्या था कि नहीं? What is the last note that you have written in the book? अरे पूरा sentence बोलो, net reaction of what? C3 cycle? ओके मुझे समझ में आ गया, यू डिड नॉट डू द C4 साइकिल क्लास। वहाँ पे तो लिखा हुआ नहीं है ना C4 साइकिल। Okay, forget, I'll explain. C4 cycle, you have attended the class or not? Bhul gaya na, haan, like you all have so many things to think about, so you generally forget. No? Iman bostu bhabibo laga ase jibonot hoy na hoy. C4 cycle korisan nai heto jodi najano keneke hobo. Understood what I said in SMS? C4 cycle का class किया था कि नहीं किया? नहीं किया था. You have to see the copy and then you realize whether you have done or not. अगर C4 cycle का class attend नहीं किया था, then what was your duty? चलो आज पूरा गाली देता है तुमको ना. अब you go home and you'll tell मैम ने आज पूरा class के सामने मुझे पूरा एकदम सुना दिया. My point is, if you did not do your C4 cycle class, what was your duty? लेक्चर देख लेते है ना, नहीं देखा ना, कौन सा लेक्चर? C4 साइकिल का लेक्चर. ओके forget, let's not waste your time. Okay, ये तो पता है कि if you are talking about dark reaction, we have to study under 3 headings: C3 cycle, C4 cycle, CAM cycle. C4 cycle, what is C4 cycle? C4 cycle, what is C3 cycle? The pathway of dark reaction that occurs in the C3 plants is called as C3 cycle. What is C4 cycle? The pathway of dark reaction that is seen in case of C4 plants is called as C4 cycle, right? So C4 plants ka example, sugarcane, maize, etc. Another word that is important, tropical plants, generally tropical monocots, right? These are the words that are important for C4 cycle, right? C4 cycle, another important thing, in this type of plant, photosynthesis occurs in 2 cells. In normal cases, in maximum of the plants, if you are talking about C3, photosynthesis occurs only in mesophyll cell. But C4 plants mein photosynthesis occurs in 2 cells, mesophyll cell and bundle sheath cell, right? Next is, if you are talking about C4, the leaves of C4 plants have a special type of anatomy. What is anatomy? Internal structure of the plant or the body is called as anatomy, right? So if you are talking about the leaves of C4 plants, they have a special anatomy, and what is the special anatomy? It is Kranz anatomy. What is Kranz anatomy? If you are talking about the leaf, no, leafot ki thakibo, vascular bundle thakibo na thake. So if you are talking about this vascular bundle, no, in case of C4, vascular bundle is surrounded by 2 rings of cells. Right? Since it is surrounded by 2 rings, no? This is one ring of cell, this is another ring of cell, and it looks like, suppose if you cut a portion, no? It looks like this one. This is the vascular bundle, this is the first ring, and this is the second ring. It almost looks like a crown, and the meaning of crown is equal to grains. You see a structure like this. So this is what is called as Kranz anatomy, right? So in the leaves of C4 plants, you see a special type of anatomy that is called as Kranz anatomy. What is Kranz anatomy? Arrangement of 2 rings of cells. What are the 2 rings of cells? One ring of mesophyll cell, the other ring of bundle sheath cell. As I said, photosynthesis occurs in 2 cells, right? So, a duta cellot ki hobo photosynthesis, it will take place, vascular bundle surrounded by 2 rings of cell. This arrangement looks like a crown, and so the name is called as Kranz anatomy. Clear? Study this portion and please bring the doubts, okay? Okay, C4 cycle or pathway to Akim. So if we look into the reactions are almost the same that we have discussed in C4 cycle, no? C4 cycle or diagram juntu akisa pathway, hetu aru etu compare kori so you have it in your notes, right? Just see. Do you find any difference? Do you find any similarity? As far as the reactions are concerned, it's almost the same. What is the difference? C4 cycle ot moi C4 cycle ot etu jetia likhisilu ei bostutu disilu nai soason PPDK na idea, please write it down. Maybe in the last class I have missed it. The enzyme that is responsible for the regeneration of PEP is PPDK, and what is the full form? Pyruvate phosphate dikinase. So it's almost the same. Okay, so what is the similarity and what is the difference? Reactions almost same, right? The products that are formed, regeneration process. If you are talking about the 3 steps of that reaction, that is carboxylation, regeneration and reduction, all the 3 steps could be seen in this pathway. C4 to dekha paise, ya to dekha paise. You know, what is the basic difference? Difference to obviously ase. Difference to ki? Pathway kita to dekha paisa, no? Pathway kita compare kori pelai kwa na? What is the difference? The difference is only day and night. Tat ki asile? Duta cell, right? Ki asile? Mesophyll cell or bundle sheath cell. Yatu moi duba differentiate kori disu. You know, what is the difference? Are we talking about 2 cells being associated with this process? No. It is the single mesophyll cell, but the condition during the night time and the condition during the daytime. And in the last class, what I have said is to write certain short points out of the pathway. I don't know whether you have written or not. Koisilu no points bilak ulai pelai likhi bole. Thik teneke in this case also, you can take out your own points, the points that we have explained. So if you are talking about carboxylation or first carboxylation occurs during ketia. Night in CAM plants, the initial fixation of carbon dioxide, we have already written that point. So the carboxylation reaction starts at night. Who is the acceptor? PAP. What is the enzyme involved? PAP case. What is the first stable product? What is the first product that is formed? Oxaloacetic acid. Oxaloacetic acid gets converted to the malic acid, and what happens to malic acid, or when is malic acid synthesized? Last class what ki koisilu? When we were talking about C4, the question regarding malic acid was, in which cell is malic acid synthesized? Etia question to moi ki hudisu, when is malic acid synthesized? Because here we are concerned with one cell itself. Kintu C4 ot ki asile? Duta cell asile. So the question was, where is malic acid synthesized in C4? Where was it synthesized? In the mesophyll cell. What happens to the malic acid that is synthesized in the mesophyll cell in C4 plants? It gets transported to the bundle sheath cell. These are the points that we have discussed. Yate ki discuss korim. Obviously, oxaloacetic acid gets converted to malic acid. Malic acid formation ketia hoy? When does malic acid synthesis occurs? Occurs during the night. What happens to malic acid during the night? This malic acid remains stored in the vacuoles. Karon ami ki jano, we have already said no. A2 combination of ki form korbo? Oxaloacetic acid get converted to ki? Malic acid gets converted to ki? Pyruvic acid. So here also malic acid will ultimately form pyruvic acid itself. Kintu since the reaction has started during the night, formation of pyruvic acid, rati hobo ne dinot hobo? It's already written no, dinote hobo, right? So jedi malic acid e pyruvic acid dinot form kore, malic acid to rati e form ho goise. So rati malic acid or ki hoy, it remains stored in the vacuoles, right? It is only when the light falls, malic acid gets decarboxylated, and ultimately what is formed is pyruvic acid. So question akko ahibo pare, decarboxylation of malic acid occurs during dash time in CAM plants. It occurs during the daytime in case of CAM plants. Decarboxylation of malic acid occurs in which cell in C4 plants? Decarboxylation of malic acid occurs in which cell in C4 plants? Bundle sheath cell, right? Kintu yate question to ki? Decarboxylation of malic acid occurs at which time of the day? Or occurs at dash time in CAM plants. It occurs during the daytime, right? When malic acid forms pyruvic acid keneke form koribo carbon dioxide ulai dibo and this process is called as decarboxylation. The carbon dioxide that was released would accepted by RUBP and the cycle would continue. What is this? C3 cycle. C3 cycle karone ki form koribo? One molecule of glucose, right? So rest of the explanation exactly the same as C4 plants. As far as the ATP requirement is concerned, the NADPH2 requirement is concerned, whatever we have explained in C4, the exact same explanation in case of this also. By now, I think we understand 18 yat use kore, baki reactions of kiman use kore 12. So what is the total amount? That is 30. So in CAM plants, for one molecule of glucose to be synthesized, what is the requirement of ATP? 30. 30 ki hor, yate 18, hetu process of 12. Right?
It will be NADPH or NADP?
NADP. I think it's clear. So points ami ki monot rakhim? When is food synthesized in CAM plants? Food is synthesized during the daytime in CAM plants. Food normally ketia synthesized hoy, rati hoy, right? Next question could be initial carbon dioxide fixation, final carbon dioxide fixation. The second question could be how many molecules of ATP and how many NADPH2. Third question could be what happens to malic acid during the night, right? Taken to already explain kori, so I think these are the points that you can just take it out and write it down, right? Any point that you did not understand, take your time and see. So up till here what we have done is we have completed the dark reaction. We have talked about C3, C4 and CAM. Is this over? Hold no. So kiba doubts ase niki? Jedi doubts nai, next heading likhiba C2 cycle or photorespiration. আৰু এবাৰ কোৱাচোন অকণ ব্ৰেক হৈছে ভইচটো PPDK থাকিব because that is the enzyme that is helping in the regeneration of PEP. Okay, so I think in the last class I have missed out this point. So তোমালকে C4 cycle or jo notes to likhisa tate ei bostu to likhilwa PPDK the same enzyme, right? Or as far as the number is concerned yete to sotai hobo aru. Any more doubts? Okay, any more doubts? Next heading kora photorespiration oblique C2 cycle. Daytime itself, yeah, because during the daytime only the complex molecule will get converted into simple molecules and this process occurs only during the daytime. So it will start. Amar janike normal plants or ki hoba, the food gets synthesized, it stays during the night and it's only in the next morning that they get broken down into simpler compounds, right? Yat tenukan hoy immediate hoy jabo, right? So many names. It is called as C2 cycle, it is also called as photorespiration, it is called as glycolate metabolism, it is also called as photosynthetic carbon oxidation cycle. Right. Out of this, normally I mean because but once the question has been asked from this point also, at this time what we are talking about is photosynthetic carbon oxidation cycle. Do you remember something called as photosynthetic carbon reduction cycle? Photosynthetic carbon reduction cycle, duta cycle ase, eta hol photosynthetic carbon oxidation cycle aru eta hol photosynthetic carbon reduction cycle. We have used this term PCR cycle code likhisilu jot carbon reduction hoy, carbon reduction code hoy C3 cycle hoy, right. So carbon dioxide loy no Rubisco will fix what carbon dioxide, but in this case since we have discussed a little bit before also, we know that this time Rubisco will take in what oxygen instead of carbon dioxide. So Rubisco if carbon dioxide loy, that process would be called as what photosynthetic carbon reduction cycle. The normal C3 cycle that we talk about, kintu yate ki likhise oxidation cycle. Okay, so you have to pay interest to that also. Okay, before writing the notes, just to have an idea about this cycle. First, why is it called as C2? Jeneke, we have an explanation now. Why is it called as C3? Why is it called as C4? Similarly, a cycle took C2 boli kyo koy because the first product that is formed is a 2 carbon compound. The same type of explanation. First stable product, if it's formed as a 3 carbon compound, tak ami ki boli kom C3, right? Yate ki hobo, the first product that is formed is a C2 or carbon 2. I mean 2 carbon compounds, so it is called as C2 cycle. Next is the word photorespiration. Why is it called as photorespiration? It is called as photorespiration because the same exchange of gases takes place in presence of light. What happens in respiration? You take in oxygen and you release carbon dioxide. In this case also, the plants will take in oxygen and release carbon dioxide, but obviously in presence of light, and so it is called as photorespiration because respiration of G gas exchange hoy. Yato ki hobo? Exactly the same process, kintu in presence of light. Light thakile hai photorespiration hobo. Jodi light na thake, then plants oxygen lobo nware, right? Glycolate metabolism, because one product that is formed is glycolate. আমি যোনটো carbon 2 বা 2 carbon compound কথা কৈ আছো সেই compound নাম কি glycolate. So we'll be talking about, and this is something that we have already understood, right? So that is the meaning of the names that I associated. Second point is C2 cycle of photorespiration is harmful for the plants, useful for the plants. It is something that's harmful for the plants, right? Harmful for the plants buli exactly ami kobo nwaru. Right, harmful, instead we use a word that is called as a wasteful process, right? Photorespiration at ki hoy, plants bilake carbon dioxide or holoni, oxygen loi loi, to oxygen jodi loi loi, tetiale photosynthesis process to ki hobo, inhibit hobo nohoy. If it gets inhibited, so obviously this would be considered a meaning that will carry a negative sense. Positive terms to ami ketiao noko, jeneke photosynthesis is a positive aspect, kintu photorespiration obviously carries a negative aspect. Why is it called as negative? Why is it called as wasteful process? It is called as wasteful process first kiho karne it does not synthesize any form of ATP or any NADPH2 during this process. Light out hoy ase oxygen loise kintu kuno tar reaction ot ATP aru NADPH to synthesize nohoy aru ATP aru NADPH to synthesize to nohoy. Apart from that, what it does is to run the reaction, it uses the ATP and NADPH that was already present in the cell. Normally, if you talk about that assimilatory power, kini ketia use hobo lagisile during dark reaction, so the food gets synthesized. But this is a process where tahate to nije nobonai, tahate ki kore cell to thaka already thaka ATP or NADPH to kini use kore so that the reaction goes on. So that is why it is called as a wasteful process. Next is another point is it is a 3-cell organelle process. So this entire reaction quote hoy tinta cellot ekeloge hoy. Right? Jeneke ami kolu C4 cycle duta cellot hoy. Thik teneke in case of C2 it's always 3 cells that are involved, and so it is called as a 3-cell organelle process. What are the 3 cells that are involved? Since it's something related with photosynthesis to chloroplast to eneo thakibo, right? So the 3 cells would be chloroplast, peroxisome and mitochondria. You have heard about chloroplast, obviously we know about chloroplast. Mitochondria is also a very commonly spoken about organelle, jot niki ATP synthesis hoy. What about peroxisome? Have you heard this term before? Hunisan nai? And if you say no, you get a chance of listening to my beautiful sentences again. Obviously you have heard, we have done cell. Cellot ami cell organelles korisilo, cell organelles ami peroxisomes, perosomes eblak korisilo, right? So one of the smaller cell organelle, one of them is a peroxisome. So etu process ketat hoy tinta cellot hoy. Now since the entire reactions runs in 3 cells, so obviously the reaction would be a long one, right? We'll be drawing that process, but we do not have to remember the steps of the reaction. Kuntu cellot arambho hoy pelai kuntu cellole products bilak circulate kore. This is something that we don't have to read. What we need to read is, since 3 cells are associated, in each of the 3 cell one important products get synthesized. Mitochondria at a particular product that has importance gets synthesized. Peroxisome at an important product synthesized or chloroplast at an important product. So the question has been asked about the 3 important products. For example, we'll be talking about something called as glycine, we'll be talking about something called as serine, right? So question to ahibo pare is a glycine synthesized kuntu cellot hoy, but serine synthesized kuntu cellot hoy. These are the points that you have to remember, right? So we'll be writing it down. Next, again, since we have talked about the plants taking in oxygen, the point is, during what or under which situation will a plant take in oxygen? Normally they have a tendency to take in carbon dioxide so that the photosynthesis process goes off well, right? Kintu oxygen ketia loy? Oxygen lobo when the amount of oxygen sometimes becomes more as compared to the carbon dioxide, right? Cellor bhitor oxygen quantity jodi besi hoy jay, Rubisco has a tendency to take in oxygen instead of carbon dioxide. So Rubisco oxygen ketia lobo jetia carbon dioxide amount in the cell would decrease. Clear? Next, no doubt we are saying that the oxygen jodi loy photosynthesis hamper hobo. But in certain plants, if you are talking about C3 plants and if you are talking about CAM plants, tahator no doubt this is a very wasteful process. Kintu jodi C3 plants or C2 cycle no hoy, then this will also have a negative impact on the cell. At one hand, what we are saying that this entire reaction is a wasteful process, right? What it does, it slows down the rate of photosynthesis. But on the other hand, what I am saying is, if this cycle does not occur in the C3 plants, then something gets hampered. What gets hampered? We have something called as photooxidation or solarization. In very simple meaning, what we know, je is a high oxygen content hoy jabo, cell tor bhitor high oxygen content hobo, he karone photorespiration hoise. So cell tor bhitor jodi a high oxygen content to oxygen khini jodi kuno loi noloy, this amount of oxygen will have a negative impact on the chloroplast. Bhangi dibo, right? So chlorophyll molecule, I think you remember we have talked about photooxidation, right? High intensity of sunlight, high intensity of oxygen, hole ki hobo, the main molecule, main structure will get hampered, aru he karone the plant is, he has to undergo the the C2 cycle. Eta side ami ki koisu, it's very harmful. Eta side ki koisu, it has to happen. Harmful in which respect? Harmful in this respect that the amount of food that is synthesized lowers down or it comes down, right? Efficiency in level to komi jay. Kintu useful keneke je jodi C2 cycle no hoy, tetiale main structure to e bhangi jabo in presence of high intensity of oxygen, right? Okay. So we'll start writing. Oh, Areta, another point. So this time ki hobo, if you are talking about RUBP, since we are talking about Rubisco, so obviously the compound that is important is RUBP only. RUBP normal case of ki lobo, carbon dioxide low pelai ki form koribo phosphoglyceric acid, and ultimately they will form one or they will form the glucose. Now if you are talking about this situation, RUBPA, carbon dioxide loi nai, ebar ki loise oxygen, tetia hole glucose molecule synthesize hobone nohoy. That is the question, right? If it takes in carbon dioxide, we know food gets synthesized. If it takes in oxygen, food synthesis hobone nohoy. Actually sabole gole hobo nalagisile. Karon carbon dioxide lole hai, the carbon will get reduced and form what? The carbohydrate. But in this case ki loise oxygen loise, so food to prepare hobo nalagisile, so it would have been a complete loss of carbon inside the plant. But we will see that as the reaction goes on, no, at a particular step, what happens? There is evolution of carbon dioxide. Jeneke ami decarboxylation process to porisilu, no, malic acid or, right? Hetu ki hoy? Intermediate reaction. Pothomote ki arambho korisile? PEP plus carbon dioxide, right? It formed what? Oxaloacetic acid. Oxaloacetic acid formed what? Malic acid. What happened to malic acid? Decarboxylation, right? So it's an intermediate step in which carbon dioxide was evolved. Similarly, in this cycle also, eta stepot ki hobo? Carbon dioxide by decarboxylation hobo. And this decarboxylated carbon dioxide ki koribo would be accepted by the RUBP and C3 cycle akko hobo. Buji paise ne nai? Right? So if you say that there is a complete loss of carbon, that statement would be a wrong statement. Normally, complete loss of carbon hobo laisil. Kiho karone? Karon plant se carbon dioxide lua nai. Kintu complete loss nohoy. Kyo complete loss nohoy? Because at one step carbon dioxide gets evolved. A jetia cell to carbon dioxide thakibo. Jitu karono karone nohoy. Either it's taking it from the atmosphere or it's a byproduct of a certain reaction. What is the last thing that we are seeing? The presence of carbon dioxide inside the cell. Whenever carbon dioxide is there in the cell, RUBP will take in the carbon dioxide and the C3 cycle will run, right? So A2 step or to food form hobo, A2 step or to food form hobo. Right? But this will be a harmful process because it will use up the ATP that is present in the cell. Plant dollar energy requirement to ki koribo borhai dibo, no? Okay, well, right. The term photorespiration was given by এইখিনিটো বুজি পাইছো So RUBP will take in oxygen instead of carbon dioxide, and Rubisco will act as an oxygenase instead of carboxylase, right? When do you see C2 cycle, or what is the condition required? The condition required is the cell should have high concentration of oxygen and low concentration of carbon dioxide. Next is it is not linked with ATP generation. I mean ATP generation of kotha to kihor karone koisu, because we have written this word called as photo. Normally in photosynthesis, whenever we are talking about photo or light reaction of ki synthesized hoy ATP. So it is almost, it is something that's occurring during the daytime itself. So light reaction type kintu tothapi ATP synthesis hua nai. Instead they are consuming all the ATP that is present in the cell, and so it could be considered as a wasteful and a harmful process, right? Next we have a word that is called as dark respiration. So what we understand by this word dark respiration boli kiyo koise? C2 cycle dark buliu koise, respiration buliu koise, because the reactions look almost like respiration. Yes or no? Karon oxygen loise carbon dioxide ulai dise, etu ki hoy? Respiration e hoy. Dark word to kiyo koise? Dark reaction or logot compare korise. What happens during the dark reaction? ATP gets consumed. Yate ki hoise? ATP gets consumed. So as a whole they have used the term that is called as dark respiration. So what is dark respiration? Nothing but the C2 cycle. Why is it called as dark respiration? Because ATP gets consumed in a dark reaction, it is getting consumed in this cycle also, right? Next, as far as this reaction is concerned, it's not important, okay? But conditions kini janibo lagibo C2 cycle hobo lagile. Next we have to draw the pathway. Is this open? लिखी लो. We have to draw 3 cells, okay? So you need to draw 3 cells. we have drawn 3 cell organelle day. Out of these 3 cell organelle, in one of the organelle you have seen a single line and the next two you have seen double line. Please draw it like that. This talks about the single membrane structure and a double membrane structure. If you are talking about chloroplast ki hoy double membrane. If you are talking about mitochondria is also double membrane. So, we have drawn it double, alright. But if you are talking about peroxisome, it is a single membrane organelle. As I have said, you do not have to by heart the reactions, right. Key question hai about 3 important products that is formed in each of the organelle. If you see in the diagram, I have marked 3 products: glycolate, glycine, and serine. So, question hai glycolate code produce hoy, glycine code produce hoy, serine code produce hoy. That is one question that is asked, right. Next is apart from those 3, no, ketiba khude H2O2 code hoy, thik ase, ammonia. So, ekita point okuman sabo lagibo kintu iman important nohoy, important ki A2 aru A2. And obviously this one, no, carbon dioxide evolution hobo. So, as I said, no, intermediate reaction eta ki hobo, carbon dioxide evolve. This carbon dioxide will be later used in the C3 cycle. Contour. This one. This one. 2 RUBP plus PCR cycle. Oporot, ekini no? This one. It will take 2 oxygen. Photorespiration ketia hobo jetia oxygen lobo no, oxygen kune lobo RUBP lo pelai ki form koribo phosphoglycolate. Phosphoglycolate pora phosphorus part 2 ulai goi pelai ki form koribo glycolate. These points are important. Ekhini par question ahe. Reactions obviously we don't have to remember. So it is mitochondria jot niki question ahibo pare. Ammonia code produce ase, carbon dioxide code produce ase. I think this cycle is over. Aru eta bostu lagibo ba. Ekhini likhil basun. Chloroplast, double membrane, peroxisome, single membrane, mitochondria, double membrane structure. Okay, so as I said, no, C2 cycle of ki hoy, RUBP takes in oxygen instead of carbon dioxide, and so we think as if there is a complete loss of carbon dioxide inside the cell. Why? Because since RUBP has not taken in carbon dioxide, tethale cell to carbon dioxide to thakiboi nalagisile, right? But what we said is there is not a 100% loss, because at a certain time ki hoy, carbon dioxide evolve hoy, and plus there is another reaction. Reaction to liquid. Ekhini likhil basun first. Ekhini ho gol niki. Is this over? This part? Nahi hua likhil ba. So the next part of discussion will be how is carbon dioxide recovered during this process. Actually, there should have been a 100% loss because the cell is not taking in carbon dioxide, but we see at the end of the process there isn't a 100% loss. Instead, there is just a 10% loss in carbon inside the cell. So 100% loss hobo lekisile kintu okol 10% to kunubai recover korile nai. Are you understanding? So the total loss should have been 100% because the cell is not taking in carbon dioxide, right? Kintu at the end of the reaction we will see that there is just a 10% loss and not 100%. Kintu as far as our discussion will be showing 75% loss keneke hoise dekham. Eto finish korilu agote. Over. Over. Done. Okay, points are under. This was the first point. Second point likha. Okay. So, as we were talking about the complete loss of carbon, but we have said that 100% loss nohoy. Kihor karone 100% loss nohoy? Because during the reaction, there are certain steps of the reaction which will go for recovery of carbon or formation of carbon inside the cell, right. One reaction we have seen that this glycine to serine, right. What we have seen in mitochondria, we see 2 molecules of glycine getting converted to 1 molecule of serine, right. Glycine or 2 molecule keita serine form korise eta, right. Glycine ki hoy 2 carbon compound, right. 2 carbon compound keita molecule 2 molecule, so ultimately carbon kiman 4 carbon, serine ki carbon 3 carbon. So, 4 carbon etai 3 carbon compound to form korise eta carbon left over hole nai, right. Hey eta carbon gets released in the form of carbon dioxide inside the cell, and this is one way in which carbon dioxide is recovered inside the cell. Carbon dioxide cell tur bhitor ekebare thakibo nalagisile karon kuneo carbon dioxide accept kora nai, kintu we have seen a reaction that occurs in mitochondria jar karone carbon dioxide form korise, form kori play kot ase cell tur bhitorte tu ase, plant or bhitorte tu ase. So, this is the recovery process. Because of this process 100% loss nohoi pelai. Ultimately it's a long calculation, we will see 25% loss hoy aru tatkeo beshi jodi ami calculate koru, question to jodi ahe what is the actual loss of carbon dioxide during C2 cycle, it is just 10%. So, ultimately 10% loss hoy, 90% e ki hoy recover hoy. Keneke recover hoy? By reactions. Aru kiba kibi hoy kintu amak iman nalage. So, just this is the point that we will remember, right. 2 molecules of glycine will form 1 molecule of serine, and in this process 4 carbon compound will form a 3 carbon compound, 1 carbon gets released and this carbon in the form of carbon dioxide remains in the cell. So, ultimately this is one way through which the plant gets in the carbon dioxide. Clear? So, this was point number 2. So, we do not go for the exact calculation as to what is the amount of loss. In general, we have to study a line, that is what is the actual loss of carbon in C2 cycle. Next point likha, point number 3. So, as I said, so no doubt this is a wasteful process, this is a harmful process, etc., etc. But in spite of that, this process needs to occur in C3 plants, because if oxygen intensity keeps on increasing, then the protoplasmic parts will get oxidized during high intensity of light. So, during the daytime oxygen বেশি হয়ে আছে, লাইটও বেশি হয়ে আছে, তেতিয়াহলে কি হবো, ফটো অক্সিডেশন বা সোলারাইজেশন হবো। আর ফটো অক্সিডেশন বা সোলারাইজেশন হয়ে গলে the main constituent will only break down, right? আর যদি main constituent break down হয়, obviously the cell will also die, right? So, in spite of being a harmful process, it has to occur. Next, photorespiration absent in quote C4 plants. Why it is absent? কারণ তাত কি হয়, কার্বন ডাইঅক্সাইডৰ এমাউন্ট বহুত বেছি। RUBP does not get a chance to bind with oxygen, right? ইয়াতে কেতিয়া হ'ব, photorespiration কেতিয়া হয়, যেতিয়া কার্বন ডাইঅক্সাইডৰ concentration কম থাকে। Whenever there is a low concentration of carbon dioxide, only then photorespiration occur, right? But in case of C4 plants, as we have already discussed, from two sources the plant gets the carbon dioxide. So, obviously carbon dioxide concentration কেতিয়াও কমি না যায়। আর যেহেতু কমি না যায়, RUBP will always act as what? Or Rubisco will act as a carboxylase, right? So, this is all about the C2 cycle. So, what we have done is we have completed the light reaction and the dark reaction, right? Dark reaction, and what we have seen that ki hoy, the food gets synthesized. As far as the different plants are concerned, the pathway of dark reaction is different. Apart from the three main pathways C3, C4 and CAM, one pathway that we see in C3 plants, to be more precise, is the C2 cycle. What happens in C2 cycle? We have discussed. Recovery of carbon hoy ne nohoy, hoy, keneke hoy, intermediate reaction hisabe glycine gets converted to serine, right? So, up till here dark reaction is completed. What next? The next part we have to talk about something called as Chemiosmotic hypothesis, right? Chemiosmotic hypothesis talks about what? It talks about ATP formation. Whenever we talk about ATP formation, we generally remember the process of respiration. Respiration not ki hoy, the food gets broken down and ATP gets synthesized, right? So, ATP formation keneke hoy boli kole, one major process involved is obviously respiration. But we have seen that during the process of photosynthesis also ATP gets synthesized. So, ATP synthesis hoise, keneke hoise, the study is called as chemiosmotic hypothesis. So, chemiosmotic hypothesis is something that we have to study. Jihetu light reaction or ATP synthesis hoise. So, the next point is ekini hogol no, heading koriba chemiosmotic hypothesis. So, we will be talking about in photosynthesis ATP synthesized kot hoy. Normally when we talk about ATP synthesis, powerhouse of a cell boli kole kot hobo, mitochondria hobo, kintu photosynthesis oto ATP synthesized hoy. So, where does ATP get synthesized? Is it in the mitochondria or is it in somewhere else? Obviously somewhere else kot chloroplast. Chloroplast jodi ATP synthesis no hoy, te dark reaction keneke hobo, right? So, we will be talking about this now. So, heading kora chemiosmotic hypothesis. So, Chemiosporting Hypothesis, the concept was given by Peter Mitchell. So, this is something that we are going to study both in respiration and photosynthesis. The process would be different, the site of ATP formation would be different, right, but the process keneke ATP form kore, the same explanation would be in case of photosynthesis and also in case of respiration, right. So, Chemiosporting hypothesis talks about what? It talks about ATP formation. We know ATP is formed during photosynthesis, it is also formed during respiration. The process, photophosphorylation and phosphorylation. What is the basic difference? In presence of light and in presence of oxygen. So, ATP forms in presence of light. A process to ami kot dekha pam photosynthesis or process to ki boli kom photorespiration. ATP formed in presence of oxygen, process is called as phosphorylation, ami kot dekha pam respiration. Next point is photophosphorylation. What you will do? Quote occur kore keneke likhim. Okay, photophosphorylation, the first point. Second point likha. Take teneke phosphorylation, the first point, second point. So, photophosphorylation occurs in yate ki likhim? What hobo? Chloroplast. Aru yate ki hobo? Mitochondria. So, Chemiosporting hypothesis talks about ATP formation, right, and for ATP to be formed tar nijor requirement ase jeneke photosynthesis hobo lagile requirement thakibo thik teneke ATP form koriboleo requirement thakibo, and what are the two requirements? There should be a proton gradient and there should be an enzyme that is called as ATPase. What we need to remember, whenever there is a proton gradient, only then ATP will get synthesized, or the other way around is whenever there is a proton gradient, only then a specific enzyme called as ATPase will get synthesized, and if that enzyme gets synthesized, only then ATP would be formed. So, ATP form koribo lagile ei duta bostu thakiboi lagibo. Ki lagibo? Proton gradient. What is proton gradient? Proton we understand. Gradient, I think you know that meaning also, because we have talked about this water potential gradient, if you remember. Gradient mane ki difference, right? You should have two area. In one area the proton amount should be more, in the other area the proton amount should be less. So, duita etat besi etat kom ki hol difference. So, that is what is called as a gradient. So, proton gradient thakibo lagibo tetia he enzyme to synthesize hobo, enzyme synthesize hole he ATP production hobo. How does the process goes around? I think we can discuss it in the next class since the time is already over, right? So, yar continuation to ami next class on. Yaloke is the concept clear, right? So, ebilak terms napahoriba dei. Again respiration or ebilak terms lagibo kintu, right? ATP formation process to kami ki boli kom phosphorylation, right, kintu in presence of light and in presence of oxygen. I think that's all for today. Thank you.